Dear students, yesterday we have seen neurons. Today we are going to see about neuroglial cells. Yesterday I was talking about the nervous tissue is made up of two important types of cells, neurons and neuroglial cells. Neurons are the excitable cells, whereas neuroglial cells are non-excitable supporting cells. In today's class, the main objectives are: at the end of the class, you should be able to enumerate all the types of neuroglial cells. You should be able to describe the functions of the neuroglial cells. You should be able to describe the components and functions of the blood-brain barrier. Neuroglial cells are the supporting cells of the central nervous system. Do you know it contributes about 90% of the cells in the CNS and 50% by volume. As I already mentioned, these are non-excitable cells. They mainly provide the structural and functional support to the neurons. Now we are going to see the entire classification of neuroglial cells. Neuroglial cells are broadly classified as neuroglial cells present in the central nervous system and neuroglial cells present in the peripheral nervous system. As you can see in this diagram or the chart, the neuroglial cells which are present in the central nervous system are of four types. These are astrocytes, ependymal cells, oligodendrocytes and microglial cells. Whereas the neuroglial cells in the peripheral nervous system are of two types. These are satellite cells and the Schwann cells. When you see the further subclassification, there are four types of neuroglial cells in the central nervous system. The astrocyte, again in turn, there are of two types of astrocytes, protoplasmic astrocytes and the fibrous astrocytes. Similarly, in ependymal cells, you have three types of cells, ependymocytes, choroidal epithelial cells and tanycytes. Apart from astrocytes and ependymal cells, you have oligodendrocytes and microglial cells. And we will see all these cells one by one. In this picture, you can make out the all important type of neuroglial cells present in the central nervous system. Can you make out the pink colored cells there? These are ependymal cells. They are having cilia. These cells are having cilia. Then can you notice a big green colored cell over there? The big green colored star shaped cell is astrocyte. That is sandwiched between a blood vessel, capillary. Can you make out the capillary and the neuronal cell body? So the big astrocyte, the green colored cell which is shown in the figure, is sandwiched between the neuronal cell body and the capillary. Apart from that, you can see light blue colored stained cells. These are oligodendrocytes, and there are also smallest cell of the central nervous system. These are microglial cells. As I told you, astrocytes are star shaped. In the microscopy, the astrocytes can be noted like this. In this histological picture, you can see the star shaped astrocyte. Now we will see the astrocytes in detail. Astrocytes, these are the largest and most numerous cells of the central nervous system, and they are star shaped, and they will fill up the extracellular space, that is the space between the neurons. And there are two types of astrocytes, as already mentioned. These are protoplasmic astrocytes and the fibrous astrocytes. The main function of the astrocyte is they will be involved in the formation of blood-brain barrier. So as I mentioned, protoplasmic astrocytes and fibrous astrocytes, I will mention few differences between these two types of cells. Protoplasmic astrocytes mainly found in the gray matter. These are large and having a thick processes and extremely branched. Whereas fibrous astrocytes, these are found in the white matter with a slender and smooth and less branched. So the cells are mainly involved in the formation of blood-brain barrier. Now we will see what is this blood-brain barrier. Blood-brain barrier is the barrier between the blood which is present in the capillary and the neurons. It forms a selective passage of blood contents into the nervous tissue. You should know what are the components involved in the formation of blood-brain barrier. We'll go to the previous picture and let us discuss. You can see there is a red colored capillary. So within that capillary the blood will be flowing, and then you can see a big neuron over there. Between the neuron and the capillary blood, what are the structures present in? Mainly you can appreciate the endothelium of the capillary, basement membrane of the endothelium, and the astrocyte. If you notice carefully, the astrocyte, as I mentioned, is a star shaped. The processes of the astrocytes, the terminal part is slightly bulged, that those are called as foot process of the astrocytes. So these foot process will be there towards the neuronal cell body as well as towards the capillary. So the blood-brain barrier is formed by endothelium, the basement membrane of the endothelium, and the astrocytes. So here also in this picture you can make out there is a capillary, and between the capillary and the neuron you can see the astrocytes. So the blood-brain barrier is formed by capillary endothelial cells, basement membrane of endothelium, and foot process of astrocytes. So this is about the astrocytes. Now coming to the ependymal cells, the second group of cells, neuroglial cells. There are three types of ependymal cells, as already mentioned. These are ependymocytes, chorioid epithelial cells, and tanycytes. Mainly the ependymal cells lines the cavities of the central nervous system. The cavities include ventricles of the brain and the central canal of the spinal cord. The question is, what are these ventricles of the brain? In brief, the ventricles of the brain are nothing but the cavities present in the central nervous system. In each cerebral hemisphere there are lateral ventricles, followed by there is a third ventricle, followed by there is a fourth ventricle. That fourth ventricle continues downwards in the spinal cord as a central canal of the spinal cord. So the entire ventricular system of the central nervous system is lined by ependymal cells. And followed next cell is a choroidal epithelial cell, another type of ependymal cell. These types of cells present in choroidal plexus. So within the ventricles there are a structure called as choroidal plexus. So within the choroidal plexus you have choroidal epithelial cells. So these choroidal epithelial cells secrete cerebrospinal fluid. And the next type of ependymal cells are tanycytes. These are mainly lining the floor of the fourth ventricle. So you should know these three types of cells and where these are present. Basically, ependymocyte is a type of ependymal cells lining the entire ventricular part of the central nervous system. Then you have chorioidal epithelial cells which will secrete the cerebrospinal fluid, and you have a tanycytes. These are also nothing but ependymal cells, a type of ependymal cells lining the floor of the fourth ventricle. The next cell which we are going to see is oligodendrocytes. The oligodendrocytes are the cells with fewer processes and smaller cells and help in the myelination of the neurons in the central nervous system. The nerve fibers within the central nervous system is also myelinated. This myelination is done by oligodendrocytes. Then another cell of the central nervous system is microglial cells. These are smallest of all the neuroglial cells and are phagocytic in nature and derived from mesoderm. So there are four types of neuroglial cells in the central nervous system: astrocytes, ependymal cells, then oligodendrocytes, and microglial cells. Out of all these four, three cells, astrocytes, oligodendrocytes, and ependymal cells, these are derived from ectoderm, whereas microglial cells are derived from mesoderm. So overall, the functions of the neuroglial cells include: they mainly support the neuron, they will be involved in the formation of blood-brain barrier, they are phagocytic in nature with the help of microglial cells. These are actually derived from the monocytes, mesodermal derivative, produces cerebrospinal fluid, forms myelin sheath, repair damaged nervous tissue, prevent the spread of impulses in unwanted directions. So these are the main functions of the neuroglial cells. Coming to the clinical anatomy, As I told you, the blood-brain barrier, it forms the barrier between the blood and the central nervous system. If this brain barrier is damaged due to injury or trauma or due to chemical injury, then the barrier will allow the unwanted molecules to enter the nervous system. Next one is a kernicterus, that is called as bilirubin encephalopathy. Blood-brain barrier is not well developed in fetal life. So, bilirubin, if it is excess, if it is present in the serum bilirubin, if it is excess, it may cross the blood-brain barrier, and that condition is called as bilirubin encephalopathy or kernicterus. The tumor of astrocyte is known as glioblastoma multiforme. It is a highly fatal tumor. It is formed, this tumor is derived from the astrocyte. Now coming to the assignments, today's assignment, you should write a short note on neuroglial cells, describe the blood-brain barrier with clinical importance, and name the part of the CNS where blood-brain barrier is absent. So that is about today's class. Thank you. Tomorrow we will discuss about the spinal cord.